Welcome to the deep dive. We are diving into stem cell therapy today. Um, you know, it really feels like we're seeing the future of medicine, and it's not just about, new treatments. It's really about the potential to fundamentally change how we approach healing from illnesses and injuries.
Yeah, that's spot on. And for you, our listener, we've gone through a ton of recent research trying to pull out the most important stuff about stem cell therapy. The core processes, the really exciting applications, and of course the challenges that come with using stem cells.
You can think of this deep dive as a fast track to understanding this complex topic. We've gone through the historical background, the latest biotech advancements, even looked at results from critical trials and new technologies that are just emerging. The idea is to give you all the essential knowledge without overwhelming you.
Yeah, and we'll be covering a lot of ground today. We'll start with the foundational discoveries, those early insights that really set the stage for this field, and then we'll move all the way up to the latest breakthroughs happening in labs and clinics right now.
So, to put it simply, we're on a mission to uncover the most important insights about stem cell therapy, its potential, and the hurdles it still faces. But first, let's start with the basics. What exactly are stem cells?
Okay, so at their core, stem cells have two really unique abilities. First, they can self-renew, which means they can divide and create more stem cells. It's like they have this endless supply of themselves. And second, they can differentiate or specialize into a bunch of different cell types in the body. That's what we call pluripotency or multipotency, depending on how many different types of cells they can become.
So it's like they're the body's reserves kind of waiting in the wings to transform into whatever specialized cell is needed, whether it's for repair or growth, right?
Yeah, exactly. That's a great way to think about it. And that's why they're so important for regenerative medicine. The potential to repair tissues or even grow entire organs is what makes this field so groundbreaking.
It sounds almost too good to be true. When did scientists start to grasp this concept? It can't be a brand new idea, can it?
You're right, it goes back further than you might think. Back in the late 1800s and early 1900s, you had pioneers like Boveri, Hecker, Maximo, and Cohnheim. Their work on how cells behave and how tissues develop was really fundamental for us to understand stem cells as we do today.
Those aren't exactly household names. So their research basically laid the groundwork for all the amazing breakthroughs we're seeing now.
Absolutely. And then there were two pivotal moments that really propelled the field forward. In 1981, Kaufman and Evans identified what we call embryonic stem cells, or ESCs. And then much more recently, in 2007, Thomson discovered induced pluripotent stem cells, or iPSCs. These discoveries were huge.
ESCs and iPSCs, those are terms we're going to need to unpack a bit more. But before we get into the specifics, can you give us a quick overview of the different types of stem cells that are important in research and potential treatments?
Sure. We usually classify stem cells based on where they come from and what they can turn into. So the main categories are embryonic stem cells, adult stem cells, then there are perinatal stem cells, and lastly, induced pluripotent stem cells, the PSCs we mentioned. Each type has unique properties and potential uses in medicine.
Okay, let's start with embryonic stem cells, or ESCs. What makes them so unique?
Well, the most defining feature of ESCs is their pluripotency, like we talked about. They can actually differentiate into any of the over 200 cell types that make up the human body. Just think, one single ESC could potentially become a nerve cell, a heart muscle cell, a skin cell, anything. For you, the listener, that means, in theory, they could provide any cell your body might need for repair.
Wow, that's incredible. So where do scientists actually obtain these ESCs?
They are derived from what's called the inner cell mass of blastocysts. Blastocysts are very early stage embryos, only a few days after fertilization. We're talking about a point before they even implant in the uterus. These blastocysts can be sourced from mice, non-human primates, and also humans. And another key feature of ESCs is their impressive ability to self-renew, allowing scientists to maintain them in a lab for quite a long time.
So how do they actually isolate those early cells?
There are two main methods. The first is microsurgery, where they use these tiny tools to physically separate the inner cell mass from the outer layer of cells, called the trophoblast. The second method involves using antibodies that specifically target and isolate the trophoblast cells, which leaves the inner cell mass behind, ready for study.
So with this incredible potential to become any cell type, what are the biggest hopes for using ESCs in medicine? What could this mean for patients in the future?
Well, the potential is really vast. Because they can differentiate into any cell type, they hold immense promise for tissue regeneration and repair. Imagine being able to create new heart muscle cells after a heart attack, or replace damaged nerve cells in someone with a spinal cord injury. The possibilities are really mind-blowing.
It's truly amazing to think about, but those are just theoretical possibilities, right? What about their practical applications right now?
Well, right now, ESCs are proving to be incredibly valuable for creating models of human diseases in the lab. Researchers can guide ESCs to develop into specific cells affected by a disease, allowing them to study the disease process at a cellular level. This is crucial for understanding the underlying mechanisms of the disease and for testing potential therapies. Plus, ESCs are essential for drug testing and development. Because they can create a wide range of human cell populations, scientists can get a much more comprehensive picture of how new drugs might affect different tissues in the body.
So it's about understanding the disease process and ensuring that new drugs are both safe and effective. Now, let's move on to adult stem cells, or ASCs. How do they compare to ESCs?
Right, so the main difference is their multipotency. Unlike ESCs, which can become virtually any cell, adult stem cells can only differentiate into a more limited range of cell types. And these are usually cell types found within the tissue from which they were taken.
So you could say they are more specialized right from the start.
Precisely. Their main job in the body is maintaining tissue homeostasis, which basically means the constant repair and regeneration that goes on within specific tissues. For example, this happens in our bone marrow, skin, and blood. These ASCs live in very particular areas within tissues called niches. We found these niches in blood, bone marrow, skin, muscle, the brain, even the heart.
And while their potential might be more limited compared to ESCs, they are still important for treating certain diseases, right?
Absolutely. Though their differentiation potential is more restricted, they've shown remarkable effectiveness in treating specific conditions. A big advantage of ASCs is that we can harvest them from a patient and then use them for autologous transplantation. Basically, we return the cells to the same person. This eliminates the risk of the body rejecting the transplant, which is a major issue in transplant therapies. And, of course, they can also be used for allogeneic transplantation, where the cells come from a donor. Their specialized nature makes them very good at becoming the specific cells needed for repair in the original tissue. Their primary role is in repairing those damaged tissues and making sure those tissues stay healthy throughout our lives.
So what are some of the most promising applications of ASCs we're seeing right now?
Well, two key areas are tissue-specific regeneration and repair. Because ASCs are found in a bunch of tissues, they can be used to help regenerate tissues that have been damaged, either by injury or simply the wear and tear of aging. Think of things like bone marrow, skin, and muscle. They're also showing great promise for treating degenerative diseases. Their role in ongoing tissue repair makes them very valuable for therapies that aim to slow down or even reverse diseases where cells and tissues are breaking down over time.
You briefly mentioned hematopoietic stem cells earlier. Can you tell us a bit more about where those fit in?
Yeah, hematopoietic stem cells, or HSCs, are a really important type of adult stem cell. They are the stars of bone marrow transplantation. Their claim to fame is their ability to differentiate into all the different blood cells and immune cells in our body. They're essential for constantly renewing our blood and immune systems. HSCs are the foundation of bone marrow transplants, a life-saving procedure for people with conditions like leukemia, lymphoma, and other blood and immune system disorders.
That's truly remarkable. Now, on to perinatal stem cells. They sound like another category related to childbirth.
You got it. They come from tissues associated with birth, such as the amniotic fluid, the placenta, and the umbilical cord. And they're multipotent, meaning they can differentiate into a variety of cell types, although not as many as ESCs. However, their range is often broader than some adult stem cells.
So would you say they fall somewhere between adult stem cells and embryonic stem cells in terms of their potential?
Yeah, that's a good way to put it. They share some characteristics with both, which makes them very versatile and potentially useful for various regenerative medicine applications. A huge advantage is that they don't have the same ethical concerns associated with embryonic stem cells, which is a big deal. Their ability to differentiate into a range of cell types makes them very adaptable for repairing and regenerating different tissues in the body.
And you mentioned earlier that they're showing particular promise for certain conditions.
Yes. Right now, we see a lot of potential for using them in cases of cerebral palsy and diabetes. Their regenerative properties and their ability to become relevant cell types make them a novel avenue for treating these complex disorders. Another big plus is their potential for allogeneic transplantation, transplants between different people. The great thing is they don't trigger a strong immune rejection. This is because they have immunomodulatory characteristics, meaning they can actually help regulate the recipient's immune system. This could make allogeneic stem cell therapies much more widely available.
That's really exciting news. Now let's move on to the induced pluripotent stem cells, the iPSCs you mentioned earlier. Can you tell us a bit more about what those are?
Sure, iPSCs are a revolutionary discovery. Essentially, we can take adult somatic cells, like fibroblasts or skin cells, and reprogram them back into a pluripotent state. This means they regain the ability to differentiate into any cell type in the body, just like ESCs. And get this, researchers have even successfully reprogrammed other adult cell types, including some types of brain stem cells and bone marrow mononuclear cells.
Hold on, so we can take a specialized adult cell, like a skin cell, and basically make it go back in time, developmentally speaking, so it can become any other type of cell. That's incredible. What's the advantage of doing this?
Well, the most important advantage is the ethical one. Because we derive iPSCs from adult cells, we avoid the ethical concerns that come with using embryonic stem cells. And the possibilities for personalized medicine with iPSCs are just huge. Imagine taking a small sample of cells from a patient, reprogramming them into iPSCs, and then guiding them to become the exact cell types needed to treat that specific patient's condition. This personalized approach drastically reduces the risk of rejection because the cells essentially originate from the patient's own body.
It's like creating a personalized repair kit tailored to each individual's needs. That's amazing. How are iPSCs being used in medicine right now?
They have several major applications. One is individualized cellular therapeutics, where a patient's own reprogrammed cells can be used for treatment. iPSCs are also crucial for disease modeling, especially in personalized medicine. By creating iPSCs from individuals with specific genetic conditions, researchers can create in vitro or lab-based models of those diseases. These models accurately reflect the patient's unique genetic makeup, allowing researchers to study the disease mechanisms and test potential treatments specific to that individual. Another important use is in toxicity assessments and drug development. The ability to generate a wide range of human cell types from iPSCs makes them a versatile platform for evaluating the safety and effectiveness of new drugs. This allows scientists to identify potential side effects early on and to guide the development of safer and more effective treatments tailored to different patients.
That's really fascinating. Now let's dive into the mechanisms of stem cell therapy. How do they actually work in the body to achieve these therapeutic effects?
So stem cells have this amazing toolkit of mechanisms. They secrete various factors and tiny vesicles called exosomes, which are packed with signaling molecules. These molecules can modulate the immune system, prevent cell death, and even exhibit antibacterial and antimicrobial properties. Plus, they play a key role in tissue repair, intercellular communication, and regeneration.
So it's not just about them becoming new cells, they're also actively influencing their environment.
Exactly. They're real team players. Let's talk about immunomodulation first. Stem cells, especially mesenchymal stem cells or MSCs, are well known for their ability to interact with and regulate the immune system. They can actually dampen down excessive immune responses, which are often harmful in conditions involving inflammation. They do this by suppressing the overactivation of immune cells like macrophages and T cells, and by promoting the release of anti-inflammatory molecules called cytokines. This is really important because it could potentially reduce the risk of something called a cytokine storm, which is a dangerous overreaction of the immune system.
So how do these stem cells know when and how to activate these immunomodulatory effects?
Well, they have these specialized receptors on their surface called toll-like receptors, or TLRs, that can sense signals of injury or inflammation in the body. When these TLRs are triggered, they signal the stem cells to release their immunomodulatory arsenal. They also communicate with nearby cells through what's called paracrine activity, where they secrete bioactive molecules like cytokines, chemokines, and growth factors. And sometimes they directly interact with other cells to influence the immune response. For example, they can prevent the activation of T cells by releasing substances like TGF1, PGE2, and HLA-G5, or by using molecules on their surface like BCM1, PD-L1, and GAL1. They can even regulate natural killer cells by reducing IFN production. Interestingly, even with pluripotent stem cells, there are signaling molecules like leukemia inhibitory factor, or LIF, that maintain self-renewal in ESCs, indirectly affecting the surrounding cells. And we've also seen iPSCs suppress the proliferation of T cells in the lab.
It sounds like there's a lot of sophisticated communication and regulation happening at the cellular level. What about the anti-apoptotic effects you mentioned? How do stem cells prevent cell death?
So apoptosis is basically programmed cell death. It's a normal process, but in cases of injury or disease, we want cells to survive and repair the damage, right? Well, stem cells can actually inhibit this cell death, which can be triggered by things like pathogens, low oxygen, mechanical stress, or radiation. This protective effect has been observed in various conditions like heart injury, neurological disorders, and lung diseases. For instance, in low oxygen conditions or hypoxia, stem cells can increase the production of proteins like HGF, VEGF, and TGF-A1, which protect endothelial cells, the cells that line our blood vessels. Other molecules like IL-6 and IGF-1 help too by increasing the production of a protein called SFRP2.
So they're not just replacing damaged cells, but also protecting the existing ones. That's incredible. And you also mentioned some antimicrobial activity. How does that work?
Right. Stem cells can fight microbes in a couple of ways. First, they can release antimicrobial peptides, or AMPs, and they might also directly interact with the microbes. They release AMPs like lipocalins, hepcidin, and beta defensins, which directly inhibit bacteria and other pathogens. They can also increase their antimicrobial defenses by producing more LL37, a peptide stimulated by the presence of bacteria that actively inhibits their growth.
Wow, it's like they have their own defense system. Finally, how do stem cells contribute to the regeneration and restoration of damaged tissues in the body?
This is a multifaceted process. First, they help regulate excessive immune responses that can actually slow down healing. Then, as we've discussed, they can differentiate into the specific types of cells needed to replace damaged tissue. And finally, they release substances that stimulate the body's own repair and regeneration mechanisms. The tiny vesicles called microvesicles and exosomes that they release play a big role in this communication and regeneration. They're like tiny delivery trucks carrying lipids, proteins, RNA, and other signaling molecules to other cells. These molecules can promote the growth and specialization of nearby stem and progenitor cells, amplifying the regenerative effect. They also influence the extracellular matrix, which is like the scaffolding holding tissues together, preventing excessive scarring and promoting new blood vessel formation. Plus, they release very soluble factors like ANGPT1, HGF, EGF, VEGF, KGF, PGE2, and IL-10, which help restore both epithelial cells lining surfaces and endothelial cells lining blood vessels.
So stem cells are really orchestrating a complex process of regeneration and protection. The field of stem cell research is always moving forward. What are some of the most recent advancements that have really stood out?
Oh, there have been some remarkable advancements lately. One key area is our understanding of how a stem cell decides its fate. We've identified important signaling pathways, like the Notch signaling pathway, and various transcription factors that control whether a stem cell self-renews or differentiates. The Notch pathway, for instance, is crucial in many different types of stem cells, including ESCs, PSCs, HSCs, NSCs, and ISCs. Other pathways like PI3K-AKT and TGF-A, as well as transcription factors like NFP, are also being heavily investigated. These discoveries are helping us to guide and manipulate stem cells for more targeted therapies.
So it's about learning how to tell these stem cells what we want them to do.
Exactly. We're also getting much better at understanding how lineage commitment and cellular specialization work. How a stem cell decides to become a specific type of cell. And research has made huge strides in understanding the epigenetic modifications involved in reprogramming adult somatic cells into iPSCs. This knowledge is making iPSC generation more efficient and reliable, which is crucial for therapeutic applications.
And these scientific advancements are leading to real improvements in medicine, right?
Absolutely. We're seeing amazing improvements in iPSC-based methods for drug discovery, disease modeling, and personalized regenerative medicine. In tissue regeneration, there are many clinical trials going on right now that are investigating stem cell therapy for a range of conditions. These include neurological disorders like Alzheimer's and Parkinson's, cardiovascular diseases like heart failure, orthopedic conditions, hematological disorders, and even diabetes.
You mentioned clinical trials earlier. Any specific updates or any new trials that you find particularly noteworthy?
There are quite a few. For example, trials using neural stem cell transplants in Parkinson's patients have shown some encouraging results, like improvement in motor function, and possibly even slowing down the disease. In cardiology, ongoing cardiac stem cell trials show less scar tissue in damaged hearts, more blood vessel growth, and improved heart function in people with heart failure. Studies using bone marrow-derived MSCs in osteoarthritis patients show pain reduction, better joint function, and even some cartilage regeneration. And HSC transplantation, of course, remains a very successful treatment for many leukemia types, often leading to complete remission and a healthy blood and immune system. And there's exciting research using iPSCs to create pancreatic progenitor cells, which can mature into insulin-producing cells. This research shows promise for treating diabetes, with some patients even experiencing restored insulin production and better blood sugar control.
Those are all very encouraging updates showing how this research is translating into real-world treatments. And we briefly touched on stem cells' potential role in the fight against COVID-19. What are some of the latest developments in that area?
Yeah, it's been a hot topic. While vaccines are still the primary weapon, stem cell therapy has emerged as a potential supporting treatment, drawing on previous successes with severe respiratory infections. Human umbilical cord stem cells, or HUCMSCs, have shown great promise in treating severe COVID-19 cases. There are many international clinical trials underway, and it seems allogeneic MSCs, especially HUCMSCs, can help by reducing lung inflammation, promoting tissue repair, and regulating the overactive immune responses we see in some COVID-19 patients. There are still challenges, of course, like recruitment for trials and variability in stem cell preparations, but the potential is there. Plus, advancements in organoid technology, where we can grow mini organs in the lab, are helping us better understand how the virus affects different tissues. This can advance stem cell research not just for COVID-19, but for future pandemics as well.
It's remarkable how quickly research can pivot and adapt to global health crises. Now let's zoom in on specific medical fields. How are stem cells being applied or explored for application across different specialties?
Well, the potential applications are vast. In regenerative medicine as a whole, the overarching goal is to restore, repair, or regenerate damaged tissues or organs using the unique properties of stem cells. The idea is to repair damage caused by disease, injury, or aging, ultimately restoring normal function and structure.
What about oncology, where the need for new and more targeted treatments is so critical?
In oncology, the focus is on using stem cells to develop personalized cancer treatments. They're being investigated as targeted delivery systems for anti-cancer drugs, both for primary tumors and for metastases. Preclinical studies in animals show that stem cells engineered to produce cytotoxic substances can shrink tumors and improve survival. They can also carry viruses that attack cancer cells and deliver nanoparticles loaded with anti-cancer drugs, potentially minimizing side effects and targeting the tumor directly. Stem cells also have potential roles in immunotherapy, boosting the body's own defenses against cancer, as well as in drug screening, regenerative medicine for cancer patients, and even in directly targeting cancer stem cells, which are thought to contribute to cancer relapse.
That sounds like a multi-pronged attack on cancer. How about orthopedic conditions, which affect so many people and their quality of life?
In orthopedics, there's a lot of research into using stem cells to regenerate damaged tissues. Conditions like advanced osteonecrosis of the hip, intervertebral disc herniations, osteoporosis, joint injuries, cartilage damage, and bone fractures that are slow to heal are all being investigated. Some approaches involve injecting MSCs, using platelet-rich plasma or PRP to enhance MSC activity, and creating biocompatible scaffolds seeded with stem cells and growth factors to stimulate tissue regeneration. Current research focuses on improving MSC isolation and preparation, understanding the signaling pathways involved in musculoskeletal regeneration, and developing new bioactive materials to support healing.
What about heart disease, which is still a major cause of death worldwide?
In cardiology, researchers are looking at stem cell therapy and bioengineered cardiac patches to repair and regenerate damaged heart tissue after events like heart attacks. Much of the focus is on using iPSCs and cardiac progenitor cells to regenerate heart muscle and improve heart function. They're also working on bioengineered cardiac patches, which are essentially cell-based structures grown on biomaterials designed to mimic healthy heart tissue. The idea is to surgically implant these patches to repair the damaged areas.
And for those suffering from neurological disorders like Parkinson's and Alzheimer's, which have such a devastating impact.
In neurology, stem cell-based treatments are being investigated for these very conditions. The main strategy involves using stem cell-derived neurons to replace and regenerate damaged nerve cells. Research focuses on various stem cell types, including neural stem cells and iPSCs that are directed to become functional neurons. The hope is that these transplanted cells can integrate into the damaged brain networks and restore lost function.
That's truly remarkable, the idea of replacing damaged brain cells. And what about skin-related problems like severe burns or chronic wounds?
In dermatology, regenerative medicine with stem cells is showing great potential for improving wound healing, treating burns, and managing chronic skin conditions like psoriasis and vitiligo. They're also being explored for anti-aging treatments in cosmetic dermatology. Tissue engineering techniques that combine stem cells, biomaterials, and growth factors are being used to create skin substitutes that promote effective regeneration with less scarring.
And are there any advancements for vision-related issues?
Absolutely. In ophthalmology, stem cells are being studied for their potential to rejuvenate the retina and repair corneal damage in diseases like macular degeneration. The hope is to use stem cell therapies to replace damaged or lost retinal cells and heal corneal injuries, offering potential treatments for vision loss. Stem cells are also being investigated in a wide range of other fields. Immunology is looking at their use for autoimmune disorders, graft versus host disease, and improving organ transplantation outcomes. In hematology, HSCs are key for bone marrow transplantation and new immune cell therapies for blood diseases. Gastroenterology is exploring their potential for inflammatory bowel disease and other gastrointestinal disorders, while pulmonology is looking at their ability to repair lung tissue damaged by respiratory diseases. In reproductive medicine, there's research on using stem cells for infertility and reproductive system disorders, and endocrinology is exploring their use in managing endocrine disorders like diabetes. Dentistry is even investigating the regeneration of dental tissues, and trauma and wound healing research is focusing on their potential for treating chronic wounds, burns, and other injuries.
Wow, it sounds like stem cells have potential applications in almost every area of medicine. You mentioned earlier that there are ways to stimulate stem cells and make them even more effective. Can you tell us more about that?
Yes, researchers are exploring various techniques to boost the therapeutic effects of stem cells. Some examples include shockwave stimulation, which has shown promise in enhancing MSC development and cartilage repair, low-dose ionizing radiation, which can improve MSC function, laser therapy to stimulate cell proliferation, electrical stimulation for nerve regeneration and neuronal stem cell differentiation, non-peptide small molecules that influence stem cell behavior, and mechanical stimulation like cyclic stretch, shear stress, and pressure, which can mimic the environment of different tissues and guide differentiation. Even low-dose gamma radiation is being explored for its effects on stem cells.
It's fascinating how scientists are fine-tuning not just the cells themselves, but also their environment to maximize their therapeutic benefits. The field of biotechnology is advancing rapidly. How are those advancements contributing to stem cell research and therapy?
Biotechnology is playing a critical role. One exciting area is exosome-based therapeutics. As we discussed, stem cells release these tiny vesicles called exosomes that are packed with signaling molecules. These exosomes can influence other cells, modulating immune responses, promoting regeneration, and facilitating repair in various conditions. Using exosomes directly as therapeutics is a promising avenue for targeted and minimally invasive treatments.
So it's like harnessing the benefits of stem cells without actually transplanting the cells themselves.
Exactly. Another incredibly powerful tool is single-cell RNA sequencing, or cRNA-seq. This allows us to study the complete set of RNA molecules within individual stem cells. This provides amazing insights into stem cell diversity and how gene expression changes during differentiation or disease. It helps us understand the subtle differences between stem cells and how those differences affect their behavior and therapeutic potential.
So it's about getting a very detailed cell-by-cell understanding of what's happening. And of course, there's the game-changing CRISPR-Cas9 technology. How has gene editing impacted stem cell research and therapy development?
CRISPR-Cas9 has revolutionized stem cell research. It allows us to make very precise changes to stem cell DNA, which has huge implications for disease modeling, understanding gene function, and developing targeted therapies. For example, we can use CRISPR to correct genetic mutations in iPSCs derived from patients. CRISPR also enables large-scale functional genomic studies to investigate the roles of many genes in stem cell behavior. And newer gene editing tools like base editing and prime editing offer even greater precision and minimize unintended effects. This opens up amazing opportunities for creating genetically modified cells for transplantation and for developing personalized cell therapies tailored to an individual's genetic makeup.
It's amazing to see how different cutting-edge technologies are coming together to drive progress in this field. And this brings us to personalized medicine, where treatments are tailored to the individual patient. How are stem cells contributing to this trend?
Stem cells are at the heart of personalized medicine. As we've seen, iPSCs from a patient's own cells can create highly specific disease models that reflect their unique genetic background. This allows for deeper understanding of disease mechanisms and the development of highly targeted therapies. Gene editing with the CRISPR-Cas9 enhances this by allowing us to correct specific mutations in a patient's own cells. And in pharmacogenomics, we can use patient-derived stem cells to see how they might respond to different drugs, personalizing treatment plans and minimizing the risk of adverse reactions. And finally, by guiding stem cell differentiation, we can identify personalized biomarkers associated with specific diseases, which helps us track disease progression and monitor treatment effectiveness.
It's about moving away from a one-size-fits-all approach and tailoring treatments to each person's unique biology. We've talked about the potential and the science. Are there any specific examples that really highlight the progress being made in translating this research into real-world treatments?
Yes, there are many ongoing and completed clinical trials showing just that. For neurological diseases, some trials are exploring injecting HUCB MSCs into specific brain regions of Alzheimer's patients, mainly focusing on safety and tolerability. Other trials are investigating HUCB MSC transplantation for Parkinson's disease. For spinal cord injuries, trials using autologous MSCs have shown some positive results, though more research is needed on dosage and delivery methods. In cardiology, we have trials evaluating UC-MSCs for heart failure and studies using a patient's own CD133 plus stem cells during bypass surgery. Oncology trials are investigating CA-MSCs for ovarian cancer and high-dose chemotherapy, followed by HSC transplantation for high-risk breast cancer. And for orthopedic conditions, autologous ADMSC therapy has shown promise for knee osteoarthritis with reduced pain and better joint function, while HUCB MSCs are being investigated for other musculoskeletal conditions.
These are really tangible examples of the progress being made, but every field has challenges. What are some of the main hurdles that stem cell therapy still faces?
There are several important challenges. One is the potential for tumorigenesis, the risk of uncontrolled cell growth leading to tumors. Rigorous preclinical testing and long-term monitoring are essential to address this. Another challenge is immune rejection, especially in allogeneic transplants. Researchers are actively developing strategies to minimize this risk. Precisely controlling stem cell differentiation is another hurdle. Variability in protocols and the risk of unintended differentiation are concerns that need to be addressed. And finally, scaling up stem cell production for widespread use while keeping costs manageable is a significant logistical and economic challenge. These are important considerations for bringing these therapies to more patients. What are you most excited about in the future of stem cell therapy? What breakthroughs might we see in the coming years? The future of stem cell therapy is incredibly promising. We expect to see even greater integration with personalized medicine, leading to highly customized treatments tailored to each patient's unique biology. We'll likely see significant progress in immune modulation strategies, perhaps using genetically engineered stem cells or new immunomodulatory molecules to prevent rejection and manage graft versus host disease. Gene editing tools like CRISPR-Cas9 will continue to evolve, enhancing the precision, safety, and effectiveness of these therapies. And the convergence of stem cell therapy and bioengineering will likely lead to the creation of functional tissues and even organs using biomaterials and tissue engineering techniques, opening up new possibilities for regeneration and transplantation.
It sounds like the fusion of different scientific and technological fields is going to unlock even greater potential in stem cell therapy. We've covered a lot of ground today, from the basic biology to the cutting-edge applications and the challenges ahead. As we wrap up, what are the key takeaways you'd like listeners to remember?
The key takeaway is that stem cell therapy is a truly revolutionary field that holds immense promise for treating a wide range of diseases and injuries. We've made huge strides in understanding these cells and developing therapeutic applications, aided by advancements in biotechnology. And while there are still challenges, ongoing research and clinical trials are paving the way for more personalized and effective regenerative therapies in the future.
What would you say were some of the most important insights or aha moments from our deep dive today?
For me, it's the incredible journey we've witnessed, from the early discoveries to the incredibly diverse applications we see today. The potential to develop truly personalized treatments that address the root causes of diseases at a cellular level is incredibly exciting.
It feels like we are at the cusp of a new era in medicine. So here's a final thought for our listeners. Given the rapid pace of discovery and our growing ability to manipulate these remarkable cells, what untreatable conditions might we realistically see addressed through stem cell therapies in the coming decades? This field challenges our understanding of what's medically possible. We've only scratched the surface today, and there's much more to explore. We encourage you to delve deeper into the sources we've discussed or to seek out other studies to continue your learning journey in this groundbreaking field. Thanks for joining us on The Deep Dive.